Hi, good morning, everyone. Those watching us through YouTube and our audience live, be very welcome. I'm Dr. Lilian Mayerhofer, and today I'll be sharing a little bit of the research that we have been developing in a collaboration with Oslo University in Norway. So I'll be sharing my screen right now. Oh yeah. Great. I hope that everyone can see the screen with this cute baby, cuter than me. So that's what you're supposed to be seeing now. The way that we will organize this is we are going through the literature and a little bit of theory first. And then after this part is concluded, we are having some room for theoretical questions or questions related to the part that I have just presented. And then we are actually digging deeper into the research that is being conducted in the university. So I'm aware that we have a very heterogeneous audience. So the way that I want to conduct things is in a way that everyone can follow up, even though you're not familiar with concepts in psychology or genetics. So we will really start from the basis, and I'll try to explain in a way that that will be clear for everyone. But again, if you have any questions and there are no dumb questions, you can just ask in our small break before the research presentation itself. So today our talk, as I mentioned, is regarding early manifestations of genetics in children's mental health and the role of maternal well-being. First, I always like to start by saying where I'm coming from. So I'm a part of Promenta Research Center, which is a part of Oslo University in Norway. And for this specific project, we are working in a collaboration with Vrij University in Amsterdam in the Netherlands. This is our brilliant team. Everyone is super hardworking. And for this presentation, I'm especially thankful for Dr. Ziada Ayuresh, the beautiful lady in the middle, because in addition of being a brilliant researcher, she's also very smart in research dissemination. So some of the figures that you're gonna see, it's actually hers that I borrowed. So the program for today, a bit of a theoretical review about behavior genetics and polygenic scores, which are the core of this research. And then we actually go deeper into the early manifestations of genetics. And then we will also have a period for discussions in which you can pose your comments or bring your questions. And yeah. Oh, we're just having a little bit of echo, so I'll just ask to turn off your audios. Yeah. Okay. So as most of you know, all of the traits that we observe in human nature, they're a combination of environmental and genetics. It's the constant discussion that we say as nurture against nature. And actually now we have reached a point in which we can measure how much is the genetic contribution in a certain trait. And when I say trait, it can be from height, weight, the color of your eyes, to more complex traits like personality or depression or anxiety. So these are traits, and sometimes I'll also use the word phenotype. They are synonyms, just so we can understand, and also be introduced to a more academic vocabulary. So nowadays, what we can do is actually measure the contribution of the environment and the contribution of genes into the development of a certain trait. So for example, if I ask you, do you think that personality, for example, some people being more of an extrovert or an introvert, is this the result of the environment where they grew up? Or is this determined by the genes that someone carries, that genetic information that you inherit from your parents? What do you think? You can actually half and half.
Both.
That's both. That's a good guess. So the way that we use to measure these kind of things is the traditional twin studies. So we have, as you probably know, two types of twins, the identical twins that are also called monozygotic twins, that for practical reasons, we say that they share 100% of their DNA. It's not exactly 100%, but to simplify a little bit things. And then we have the dizygotic twins, in which even though they share the womb, they have the same gestational period, they share only 50% of their DNA. So it's the same amount, for example, that you share with your older sister or with your younger brother. It's the same. So what we do in this case is we observe the similarity in a certain trait between the monozygotic and dizygotic twins. And we also have adoption studies in which, for example, monozygotic twins grew up apart. So you can actually see that even though they are identical in their genetic content, they might show a different phenotype, a different trait, and that we would assume would be attributed to the environment, for example. So that's, in a simplified way, a basic explanation of what a twin study is. And then we have a key concept within genetics and twin studies, which is heritability. Heritability is defined by the proportion of a trait variation that can be attributed to genetic variation. And this is a number that goes from 0 to 1. So in other words, we know that there's variation in a certain trait. For example, some people are taller, some people are shorter. So what explains this variation? What is the proportion that is explained by genetics? That's heritability. So a classic example is the number of fingers in your hand. So we know that adults, in a general, we have 5 fingers in our hands. But there's variability in the number of fingers in the hands. Some people have 4 fingers, some people have 3, some people have 6. So what do you think is the heritability of the number of fingers in your hand? Is it closer to 0, meaning that genetics almost don't play a role? Or is it close to 1, meaning that the variability is mainly explained by genetics? What do you think? Yeah. So we had some different responses. So let's think a little bit together. What do you think is the cause of variability in the number of fingers in adults? We would think that it's mainly due to accidents, to trauma, to car accidents, to something that happened and suddenly you chop your finger and then you miss that finger. So it's mainly due to environmental causes. So since it's environmental causes, the contribution of genetics is just very little, because there's this trade-off. If I increase the environmental contribution, I'm decreasing the genetic contribution. It's this constant trade-off. So if something is better explained by the environment, then the genetic contribution actually decreases. So in this case, it would be closer to 0. And you might remember that in 2013, it was famous, the case of Angelina Jolie, which she did a genetic investigation, and then she tested positive for a change in a certain gene called the BRCA1 and 2, and this gene is associated with breast cancer. So in order to prevent developing breast cancer, she has opted to undergo this bilateral mastectomy, meaning that she removed both breasts to prevent the development of cancer. So it's very common to have this idea of one gene linked to a certain trait. So we would have also, for example, a presenilin gene linked to early Alzheimer, or we have, I don't know, a certain gene for eye color. So in the common sense, we have this idea of one gene for a trait. The problem is when we come to complex traits, like personality, like depression, like anxiety, like happiness, for example. In these cases, we do not have one candidate gene that will explain that behavior. So this is a so-called Manhattan plot, and you see in the middle a dotted line. And then these things that are colorful, that are blue and red, these are candidate genes that try to explain major depressive disorder. This is a meta-analysis that was published in 2019 in the American Journal of Psychiatry. So what I saw is that all of the candidate genes, like I think that this X gene is linked to major depressive disorder. All of these genes that I thought could be associated with the major depressive disorder, they are below the significant level, meaning that they are actually not significant. So they cannot explain these complex traits. So what do we have to explain these complex traits if it is not a single gene that I can attribute? Then we enter in a little bit of a more molecular level. If you remember in your high school biology, we have our chromosomes, and then we have this double-stranded DNA, which will carry our genetic information. So one strand you inherit from your mother and the other from your father. And these strands, they are linked together by this base of pairs that we simply represent by letters. So these letters, we are going to call them alleles. And they are basically a representation of your genetic information. They are another way of representing, let's say, your genes. But they are not really your genes. They are just a teeny tiny bit of your gene. And then what we do have is that the majority of people, they have the same order of letters. But it might be that in some cases, instead of having a certain letter, one person has another letter. So in this case, for example, instead of having a C, the person has a T. So that's what we call a single nucleotide variant, because these letters are also called nucleotides. And another way to call it is single nucleotide polymorphism. This is a category that arises when a certain variability is present in at least 1% of the population. So if 1% of the population has a T instead of a C, then I'm calling it a single nucleotide polymorphism. But this is a very long name, so we'll just shorten it into SNP, which is SNP, single nucleotide polymorphism. And from these SNPs, we can actually calculate the polygenic scores. So here we have 2 people. And then we have a SNP. Instead of having an A, 1% of the population has a G, for example. So that's called a SNP. And then what I'm going to do is, instead of just assessing one single place, I'm assessing tens, hundreds of thousands of places, because it's not just one. It's really big data. It's a lot of information. So let's say that I'm evaluating, for example, depression. And then I learned that having a T in a certain position is linked to depression. And then I'm taking your genome, your genetic information. And if you remember, I said that you have 2 strands in your DNA, which means that you have like 2 slots for having a letter. So let's say that for a T, I'm assessing your genome, and then in one strand you have a C, and in the other strand, you have a T. So you have a genotype score of 1, because from these 2 options, you have one that matches the nucleotide that is associated with that trait. And then I will weight this allele, because not all alleles are equally associated with a certain trait. They have different weights. So I need to weight that. And then in the end, I'll have the weighted genotypic score. So let's take another allele. Let's say that C is associated with major depressive disorder. And then in that specific place, you have a G and then another G. So you have no C, which means that your genotype score is 0. So if I multiply 0 by 0.004, I have 0 because you have no allele that will favor you to develop that certain trait. Let's take a third example for the allele A, which is associated with major depressive disorder in a weight of 0.003. Then let's say that you, Claudia, or you, Karine, you have in one strand you have an A, and in the other strand you also have an A. So we have all of the alleles that are associated with that trait. So we have a genotypic score of 2, and then I multiply and get a weighted genotypic score of 0.006. So I'm not gonna go through the hundreds of thousands of SNPs. I think you got the idea. But the point is, I'm gonna do, not me, of course, we do everything with the computer. So we'll repeat this analysis for thousands and thousands of times. And then in the end, we'll have our polygenic scores. And it's interesting to notice that I was saying major depressive disorders, but we actually have polygenic scores for basically everything. We have polygenic scores for educational attainment, meaning how long you're gonna go in your education. We have polygenic scores for smoking, for drinking alcohol. We have polygenic scores even for whether you are just living with your partner or getting married. So it's quite a lot that we can assess with these polygenic scores. So I would guess that the next natural question that you might be thinking is, but Lilian, how do we know that the allele T or C or A is associated with major depressive disorder? Because I put here on the table, I said that, I hope you believe me, but we need to show some evidence. So the way that we do that is through different consortia. For example, the 1000 Genomes Project, or very well known, the Psychiatric Genomics Consortium. So what this consortia do is they recruited many people, and then they take the genetic information of the people they have recruited, and then they also assess certain traits of these people that they have collected genomic information about. So let's say that here in this room, I'll recruit all of you. I'll take a blood sample so I can analyze your genome. Of course, it won't be the whole genome. I don't need that. It's just a small bit. And then I'll also apply some psychological tests for you, and also some social demographic questions. For example, your education, your income, your marital status, your parity maybe, and then some tests regarding your personality, regarding your degree of quality of life. And then I can see an association between the genetic information and the self-reported data that you are providing me. So these things are commonly called GWAS, which stands for Genome-Wide Association Studies. So we are just gonna call them GWAS from now on. So what we do have with GWAS is that at first, we could not really recruit that many people. It was hundreds of thousands. And then we did not have enough power to really detect all of the SNPs that were associated with a certain trait. But more recently, we have been recruiting more and more people. So the more people we recruit, the more power we have to detect these bits of DNA, these SNPs that are associated with a certain trait. So now we are talking about hundreds of thousands, even millions. And then we have the genotype, we have the scores on a certain trait or even a diagnosis. And we see the association between the genotype information and the score in the certain trait. So just for you to have an idea of how the things have been progressing over the years, let's think about the genetics of education, meaning the educational attainment. So we had our first GWAS in 2013, in which 120,000 individuals were recruited to run these GWAS. Then in 2016, we had another GWAS assessing basically the same thing, but then it was recruited 300,000 individuals. It was almost 3 times. And then we had a more recent GWAS that comes from 2018, in which 1.1 million individuals were recruited. So we are really recruiting more and more people, and we are actually already drawing from cohorts that already exist and making this sort of MEGA or meta-analysis. And with this increase in the sample size, we have an increase in power. And we started with an average of polygenic scores explaining 3% of the variability. But with the increase of power, we have it explained up to 16% of these differences. In a general, polygenic scores, they still explain just a tiny bit of the variability that we see. It doesn't explain the same amount that we see, for example, in the heritability studies. There is something called the missing heritability, but that I'll talk a little bit more in our talk on Friday, so you're all invited. Yes, I won't give you any spoiler alert. Yeah, so for now, it might have been a little bit too in-depth, but I want you all to take a deep breath, and I'll be taking questions for this part in our YouTube channel before we actually go to the applied part of this polygenic scores. So if you have any questions, please post in our chat while I'll just have a sip in the water. And also here in the audience, people in real life, you can also post questions if you have any. And just one detail, if you are maybe uncomfortable in writing comments and questions in English, that's also perfectly fine. You can do that in Portuguese, and we will try our best to try to translate your question. Okay, so I hope you all have had a small break for a little bit of water, grab a cup of coffee, and now we move forward. So now we get into the juicy stuff, which is the early manifestations of genetics in children's mental health and the role of maternal well-being. If you remember from what I just presented, usually these GWAS, they are conducted in adults. So I can see the association of a certain trait and the genetic information in adults. But one thing that we are still wondering and that we lack information about is if a child has this polygenic score or has these certain SNPs that are associated with a certain trait, let's say ADHD or schizophrenia or anxiety, how does disease manifest themselves at an early age? Because at an adult age, we kind of have a better idea. But what is the early manifestation of depression? What is the early manifestation of schizophrenia? That's something that we are curious to understand. So the questions in this research are how different polygenic scores, now we will just use PGS as an abbreviation, impact toddlers' mental health, and how maternal well-being could impact this association. So first, some definitions. What do I mean by mental health in this context? To evaluate mental health, I'm using the preschool version of the child behavior checklist, which was applied to 3-year-old children. The child behavior checklist, just for you to have an idea, it's a questionnaire that is fulfilled either by the caregiver or the teacher, and is composed by 100 questions. So examples of these questions are, the child acts too young for the age, or afraid to try new things, or hit others, or cruel to animals. So it's 100 questions that the caregiver or teacher will respond. And then, based on these 100 items, we can divide these items into different clusters. And these clusters will represent that the child is emotionally reactive with 9 items, or anxious and depressed with 8 items, or has some somatic complaints with 11 items, or withdrawn, like doesn't like to take part into activities or this kind of things, or sleep problems, or aggressive behavior, or attention problems, or other problems. So another way for us to organize all of these complaints is again by clustering them. So if we take these 4 first items, emotionally reactive, anxious and depressed, somatic complaints, and withdrawn, we can call this group as internalizing problems. On the other hand, if we take aggressive behaviors and attention problems, we can call these externalizing problems. So from now on, we'll be working with these 2 big groups, these 2 big clusters of internalizing and externalizing problems. And now you know what they mean. There are many ways to assess well-being. We have a social well-being, we have psychological well-being, we have spiritual well-being, but one very popular way of evaluating well-being within this happiness field is the so-called subjective well-being, which was brought by Ed Wiener, more specifically in 1985. So the subjective well-being is a combination of satisfaction with life, or life satisfaction, and positive affect, which we could translate as joy, for example, having joy in life. So to evaluate maternal well-being, in this case, we are using the satisfaction with life scale. And this is an example of the satisfaction with life scale. It's a Likert scale with response options from 1 to 7, from disagree completely and agree completely. And the 5 questions are: in most ways, my life is close to my ideal. The conditions of my life are excellent. I am satisfied with my life. And so far, I have gotten the important things I want in life. And if I could leave my life over, I would change almost nothing. And then these are the polygenic scores, the PGS, that we have included in the model. So we have PGS for subjective well-being. We have for major depressive disorder. We have for attention deficit and hyperactive disorder, ADHD. We have for autism spectrum disorder. We have for anxiety. We have for educational attainment. We have for schizophrenia, bipolar disorder. And where did I get my participants from? So we have a Norwegian cohort called the MOBA, which stands for Mother, Father, and Child. And it's a very long cohort. It started in 1999, and it recruited over 114,000 pregnancies. The cohort followed these women for over 20 years, and it's actually ongoing data collection, but with the children of MOBA. So the oldest MOBA child is a man, now not a boy, who lives in Bergen in Norway, and he's currently 24 years old. So when it comes to genetic material, we have genotypic information of 25,000 children. Then we excluded those who took part in a GWAS. This is a very, very important step, because if you remember, the GWAS usually draw data from other existing cohorts. So if we take participants from our cohort who also took part in the GWAS that we are using, it's like a double dipping. So to have more reliable information, we need to exclude these participants. And then lots of them didn't take part in the 3-year-old wave. And some of them had missing information in either the CBCL or the satisfaction with life scale. Since this is a cross-sectional study, we have excluded those with missing information to avoid imputation since it's cross-sectional. Then we were left with over 14,000 children that were included in the study. It's also worth to mention that these also represent unique families. We don't have siblings, so these are kind of independent children, these 14,000. And this is an idea of the basic model. This is a little bit of psychological language, but it will be quite intuitive for you to understand. So in the round, we have internalizing and externalizing problems. We just have them round due to methodological reasons because they're latent, but I want to explain, I want to get into this in depth. You can take a look afterwards. And then we have on the upper part, we have the different polygenic scores that will try to load or explain or predict these phenotypes, these traits that we are observing. So that's basically what we test. How can these different polygenic scores predict these traits that we are talking about? And then when we run this model, and an extension of this model is how maternal well-being could moderate, could change this prediction or this association. So when it comes to result, we see that externalizing problems, meaning attention deficit and aggression, can partially be explained by subjective well-being, polygenic score, ADHD, and educational attainment. So some things that I wanted to think now is that the best way to predict or to explain psychological traits is actually through educational attainment. We can predict better, for example, depression with your genes or your SNPs for educational attainment than actually for your genes or SNPs for depression itself. So you can take a minute to think, why do you think that might happen? Or I can give you a spoiler alert and say that if you remember, GWAS rely on millions of people. And then we need kind of reliable information. And educational attainment is something that it's kind of universal. A PhD is a PhD here, is a PhD in Norway, it's a PhD in China. But when it comes to having a definition or even an understanding of depressive symptoms or anxiety symptoms, things can be a little bit more tricky. So maybe this variability in the understanding of these concepts that we are measuring could explain why educational attainment is actually one of the most reliable measures we have. So as you can see, educational attainment predicts negatively both internalizing and externalizing problems, meaning that the higher SNPs you have for educational attainment, the lower chance you have for showing internalizing and externalizing problems at the age of 3. And the same with subjective well-being. One might think that subjective well-being is only linked to happiness or well-being, but actually your SNPs, let's say your bits of genes that are associated with happiness, they can protect you against externalizing problems. So against aggression and attention deficit. And on the other hand, ADHD will predict positively externalizing problems, which would be kind of what is expected. And the interesting part is when it comes to maternal well-being, because we might think that genes are deterministic. If you have the genes for something, you will necessarily develop that kind of trait that is linked with that gene. But that's not true. We are talking about the gene-environment association. So overall, anxiety PGS cannot predict internalizing or externalizing problems. But it can actually predict depending on the gradient of maternal well-being. So if the child has his or her upbringing in an environment where the mother is not happy, then the genes can actually have a role in developing, internalizing, and externalizing problems. So one thing that you might be thinking is, well, maybe maternal well-being is also influenced by maternal genes. maternal polygenic score for well-being. And the mother genes will also influence the child's genes, because everything is inherited. So what if actually this explanation is not by the environment, by actually the degree of happiness of the mother, but it's an indirect effect of the mother's genes that would cause this moderation? So we have actually tested whether this moderation would be explained by the children's polygenic score for well-being, or even by the mother's polygenic score for well-being. And what we have found was that there is a complete mediation pathway, meaning that it's not the genes, the maternal genes, that will explain this moderation, but it's actually the genes through the maternal well-being, meaning through the maternal phenotype. So this is what we have tested, whether the maternal well-being would have an effect due to maternal genes, or even due to maternal genes that are passed to the children. And then it didn't work, meaning that it wasn't significant. So the effect is really through the environment, through the degree of happiness from the mother. So as a summary, because I have been talking too much, we see that polygenic scores for subjective well-being, ADHD, and educational attainment predict externalizing problems, and that polygenic scores for educational attainment also predicts internalizing problems. And that polygenic scores for anxiety overall does not predict internalizing or externalizing problems. But however, there might be a prediction depending on the gradient of maternal well-being, meaning that the genes, or the SNPs in this case, they might act depending on the environment, in this case, the maternal well-being. So what are the implications? That we might have this common sense that if you have this genetic predisposition, you will necessarily develop a certain trait, and there's nothing you can do about it. But actually, environmental variables could be protective, even in the presence of genetic predisposition. So even though a person has this genetic predisposition, there's still room for environmental change, or, for example, public policies that could impact this association. What are the extensions that we want to do with this project? As I mentioned earlier, this is a collaboration with Vrije University in Amsterdam, and they really have very, very good genetic data from the Netherlands Twin Register. And then the idea is to include their data to have a meta-analysis. We have already received the approval for getting the data. Now we're just waiting for the package to include in the analysis. And we are also expanding our data from MOBA, because if you remember, I said that MOBA recruited over 114,000 participants, but we only had genotypic information over 25,000 children. But now we are receiving genotypic information for everyone. So we have a research assistant that has been working very hard to calculate all of these new polygenic scores from this new data package that we have received. And now we are opening for questions and discussions, anything you might be wondering. And thank you for watching.
We have a question from someone from the audience.
Okay.
Vitória Lima.
Oi, Vitória.
She's asking, send us the question is, sleep problems might affect internal and external factors. Why is not taken into consideration to define the clusters?
So when it comes to sleep problems, I think you're absolutely right that it could definitely impact both internalizing and externalizing. But the way that these clusters were created, it was not really based on a clinical observation, but rather in the psychometric properties of the questionnaire. So what they did was actually a factor analysis to explore the loadings and how many factors there should be created. So I agree with you that from a clinical and empirical perspective, it should be included. But when it comes to the statistical and mathematical models, things were a little bit different. So we opted for following the instructions that were provided in the manual, along, of course, with the psychometric information of the test. But, thank you for your question. That was very interesting.
I have a question.
Yeah.
What kind of variability do you have in Norway when it comes to educational attainment? Because my view is that most people will study at least until they're 16 or 18. How much variability is there?
Yeah. That's a very good question. And first, I should say that the variability first depends on the generation we are talking about. Because nowadays, whenever we think about Norway, we might think, oh, a rich country where everyone has access to education. But we should remember that Norway only actually became, let's say, a rich country in the 80s because of the oil money. So before that, when it comes to the public policies that would allow everyone to attend education, it wasn't that great. So when it comes to previous generations, there was indeed a greater variability among for educational attainment, and the strata, the layer that could actually take part into higher education was smaller. But nowadays, with oil money and smart public policies, there is actually a smaller variability when it comes to educational attainment, and the access has really broadened itself.
So for this generation, starting in the year 2000, right, 20 years ago,
Yeah,
right?
yeah, 98.
So this generation of parents, how much variability do you have there? So these people who were around 25 or 30 in the year 2000, they were highly schooled, I would imagine.
Yeah. And this is actually a problem with MOBA because we are still talking about a weird sample. There's still a wide, highly educated with high access to all kind of services. And actually, when it comes to using this kind of data for genetic information, we have another sort of bias because we have mainly European ancestrality, so we don't have good sensitivity for for generalizing our findings and making them applicable for example, Latin or African or even Asian populations. But that's again a spoiler alert that I will go more in depth on Friday. But I think this is a very interesting discussion. Thank you for the question.
Yeah, because when we talk about a country like Brazil, variability is much larger, right?
Mm-hmm.
You will still have today, for example, I have a sample that I collected 5 years ago, in which I have parents who have on average 3 years of schooling, and then others that have 60, 20 years of schooling.
Yeah.
And then when it comes to, it was interesting your point, because when it comes to a questionnaire such as this one, people, so you are working with the perception of the parent about their child. Answering a questionnaire which depends on how well they know the language and how well they can answer.
Yeah.
And also the mixture between how can you not separate educational attainment from social economic status and all the impact that arouses. So this is really complicated to try to mix data, which is much purer. I wouldn't call it a disadvantage or a limitation. It's just a characteristic of your sample, which is good because in one way it's more stable. But then to extend this, it's really, really difficult. It's kind of fun.
I see your point, and I totally agree with you. But on the other hand, I think it could also also give us a better clue or a better information of the impact of the environment. Because if we have, for example, someone with a very, very high polygenic score for educational attainment, or a population with very high score for educational attainment, and in practice, this population is still not reaching like the genetic potential, then something is like more wrong than we would assume at first. And there are even some futuristic discussions on, for example, in the future, whether your tax, whether how much tax you pay should actually be based on your genetic potential than actually how much you earn in practice. But this might be a little bit futuristic, or we might actually get there.
Yeah, I have the impression that when we start studying people from countries such as Brazil, the association between, for example, genetic scores and intelligence, etc., will change completely.
Yeah.
Because when you give everybody the opportunity, then you can see the genes appearing. And here you don't. Not everybody has the opportunity. Therefore, you will not see them at their full potential because they cannot go to school. Therefore, I think it will mix up the literature a lot in this respect as well. It's really important to extend these findings to other socioeconomic situations. It will probably change a lot.
Do you know any similar study in Latin America, for example?
So we do have some duos in Latin America, but I wouldn't know to tell you whether we have here, for example, identified the same SNPs that are associated. But now recent efforts have really been done to combine everything. So even though, as you very well pointed, there is a variability based on the economical context in which we live, there are still some that have, for example, the higher polygenic score for educational attainment and reach a PhD and postdoc, and we can benefit from this kind of information. Yeah, benefit in numbers, I would say.
We also have similar studies with fathers.
So we're just granted genetic information for fathers. So a very powerful tool that we have is actually the genetic information of trios, because we should remember that children, they will inherit half and half from the father and the mother. So some things that maybe the mother don't explain, the father is there. We should always remember the father. The only problem with using the fathers, at least when it comes to MUBA, the cohort that we have been working, is that the information regarding fathers is way more limited. Whereas, for example, we have many data points for women with fathers. We have one data point, which is when the woman was pregnant. And then we have another one that was collected in 2015. And then we have different fathers with children in different ages. So it's a little bit more tricky to work with. And the sample size is also very shortened. So we do want to work with fathers, but considering that the information it's quite limited, we'll get there.
Yeah, no, I'm asking because like when it comes to studies about parenting, we also have more mothers.
Yeah.
Right? And I think it's so important nowadays to include the fathers, also thinking about the narrative, the implication, right? That the influence of the mother. So that's why I was curious, because at least like I work with a different perspective, but it's always hard to kind of to recruit the father, and then we talk about even in Norway, right? Even in Norway. So we talk about parenting, but most of the time we're talking about mothering. It's not parenting.
Yeah, exactly.
So I was just curious how it is when it comes to genetics. Study.
Yeah, I would say that with fathers, it can even get trickier, because sometimes you have a father and a family is something that is very dynamic. So women get divorced, women get new partners, women get other men or other women that will help with the upbringing. So it's really a complex and dynamic structure that would really require more data to control for all of these dynamic transitions. But one thing that we were curious about, that we might not have very impressive longitudinal data when it comes to fathers, but we do have information about things that are more stable, like personality, and we also have the genotypic information. So a parallel project that we have been working on is regarding assortative mating. So what does it mean? It means how your genetic information, how my genetic information, Lilian, will determine the traits that I search for in a partner. So if I have, for example, high polygenic scores for well-being, would I also search for a partner that also has high polygenic scores for well-being? Would I search for a partner that has lower polygenic scores for neuroticism? So how can genes implicate your choice of partner when it comes to that? So that's something that we are very curious about, and we have data to investigate that. So it might be the topic of a further conversation.
Yeah.
Yeah.
How about sex? Oh, sorry. How about sex differences in boys and girls of these children? Did you have a look at them separately too?
Yes. So we do have that when it comes to the prediction of the polygenic scores, it's quite similar among boys and girls, except that the anxiety polygenic scores, they have a higher effect in women. And
Girls, you mean?
in girls, yeah, I'm sorry, in girls. And what we do have is also the prevalence of these of those that are above like a clinically significant level. We would say that the boys, they more commonly have more externalizing problems, whereas girls have more internalizing problems. Yeah.
So it's the pattern that you see later already at that age.
Yeah, already at that age. And I would even say that there's also a small difference. I haven't included here to not be too much, but when we are talking about boys, we also have a statistically significant effect of schizophrenia polygenic scores loading to externalizing or predicting externalizing problems, which doesn't happen in girls. So that's something that we could also elaborate or discuss.
That's really important data.
Yeah.
You know, follow these kids, see the ones that are more susceptible to develop that sort of thing.
Yeah. Yeah.
Oh, that's super interesting.
Super interesting, right? And we had another question.
Yes, just adding some comments. I work with paternal epigenetics of inheritance of epigenetic patterns for autism.
Oh.
And sometimes we see that families are very dynamic, and sometimes the behavior of the child is more similar to their fathers of their mothers. So we always talk about study parents in a father point of view too, because of these characteristics.
Yeah, that's indeed very important.
Biological fathers, you mean, right?
Yes.
Big things, biological. Yeah, the children are closer in behavior to their biological fathers.
Yes.
That's quite interesting. We also have some epigenetic data that we have been calculating. Just to clarify to our audience, we have two main kinds of explanation to traits when it comes to, let's say, genetics. We have the genotypic information, in which really handles with genes and SNPs and all of the things that I have been explaining. And then we have the epigenetics part, which we had a comment and very interesting contribution from the audience. in which you don't really change the structure of the DNA, or you don't really change the gene, but you change kind of the structure that is surrounding that gene or that SNP. So for example, the histones or things that can promote that the gene will be expressed, for example. So when it comes to epigenetics, we wouldn't talk about polygenic scores, but we have something different. We could have even like a polymethylation scores, but this is slightly another topic. We have been investigating the impact of epigenetics in well-being. I have been mainly working in well-being and the controlling for some environmental causes that could also have an impact in epigenetics, especially smoking, which has been shown to have a great impact in epigenetics. And in this case, really well-being is more determined by genetics than epigenetics in this case. I would be curious to see the epigenetics contribution to this study, for example, when it comes to internalizing and externalizing problems. But we still need the data, and we have been working very hard to acquire that.
This is something you can extract, right, from all the data, from especially the children, as they're very young, and we'll have so many changes there. But as time goes by, it's fantastic data.
Absolutely.
Pity about the farmers, really. It's funny, isn't it?
Yeah.
A recent study, I mean, it's set up 20 years ago, and still this wasn't in people's mind, right?
Yeah. And for me, it's even a bit surprising because the first data point actually recruited fathers, so I mean they were already there. Yeah. So yeah, it's surprising that there wasn't a follow-up, but I'm sure there were other things involved that we might not be aware of. Okay, so if we don't.
No more questions.
Oh.
No, there's no more questions.
Yeah. Okay, so as a reminder, I'm also available on email, so I'll put there again. If you wanna talk a little bit further, have any other specific question, you can always just drop me a message. I'm usually very available.
And also you'll be together
And on Friday, tell them about.
Oh yes, so now it's the advertisement part. So
And tomorrow is the one in Portuguese.
yes, so we are having a second talk tomorrow at 4, from 4 to 6 Brazilian time. And this talk will be in Portuguese. We will again go a little bit on polygenic scores. But the idea for tomorrow is to understand the maternal transitions that happen from pregnancy to toddlerhood. So how maternal well-being, well-being, and how happy the mother is with her partner, how the things will change throughout pregnancy to toddlerhood. So we are all invited. There's a YouTube link in the same channel, and we also have other very interesting talks.
Yes.
Today at 2. We have another brilliant researcher, Karina Viana, who will be talking a little bit about child development. So is it the same link? No, it's a different link.
It's a different link.
Yeah.
We could add them to the description of the YouTube channel.
So it's a different link. You see it there. And also tomorrow morning, we have a very interesting talk about genetics of musicality. You know, we are a very musical country, so it's quite interesting to see the biological and genetics basis for that. So you can have access to the full program in the YouTube channel, if not yet, soon. And you're all invited to take part. And thank you for hearing.
Thank you, Lilian.
Yeah.
Thank you so
Thank you.
much.